Welcome to another guided tour histology lab. Today we're going to look at nervous tissue. Specifically, we're going to focus on peripheral nervous system, that is, as opposed to the central nervous system, which consists of the brain and spinal cord. However, we will be taking a look at some spinal cord neurons in a smear, as well as some spinal cord sections, in order to get an overview of the various types of neuronal cell bodies. So as a general rule, any group of neuronal cell bodies outside the central nervous system is called a ganglia. We're going to look at three different types of ganglia, but before we do that, we're going to look at neuronal cell bodies from the perspective of morphology, specifically the number of projections that they have coming off of them. The first slide we're going to examine is the slide of spinal cord neurons. This is not a section, but a smear of neurons that were taken out of the spinal cord. These neurons are what we call multipolar. That means they have a number of projections, usually multiple dendrites and only one axon. Although at low mag you can't really see much of anything, as we zoom up into medium magnification, you begin to see these kind of background dots, which are the nuclei of the glial cells, and these very large cells here with numerous projections coming off of them. And those are the multipolar neurons. So let's bring one of those into the middle and zoom onto it and take a look at it. Now, because these are whole cells in a smear, you don't have the clarity or the resolution that you might expect from a section, but you can see enough to really understand that they have many projections coming off of them. So here you can see this very large cell body, and here is the nucleus with a prominent nucleolus. That's a little hard to distinguish there. There are many background glial cells in this area around it, but you can see here many cell projections, probably close to 7 or 10 projections coming off of this cell body. Here's another example from the same specimen. Here, in this case, you can see the nucleus a little bit more clearly with a really prominent nucleolus there in the middle. And you can see that you have 1, 2, 3, 4 projections visible within this frame that you can spot coming off of the soma or the cell body here. Another feature I'd like to point out on this slide is this purplish color in the cytoplasm. And when you have a smaller, somewhat smaller cell like this one, you can see a little bit clearly that these purplish areas are in clumps, and that is what we call nissl substance. That's just another word for clusters of rough endoplasmic reticulum, because these neurons are extremely synthetic cells. They're always making things, lots of proteins to maintain themselves, as well as neurotransmitters, and so on. Locating the axon hillock might be a little bit more challenging. In general, when you're looking for one of those, you want to find an area like this where the nissl substance is not present in a projection. So this is what I would think of as the most likely candidate as the axon coming off of the soma here, because there's very little nissl substance right in this area. Another type of structural neuron is called pseudounipolar. These are primarily seen in the dorsal root ganglion, which is shown here. These pseudounipolar cells are typically large to medium sized, and their cell bodies appear quite round, whereas the multipolar ones are very variable in shape. So at slightly higher mag, you begin to see other features of these pseudounipolar DRG neurons or ganglia. Let's go up in magnification, and here at a higher magnification, in this section you can clearly see the outline of the nucleus with a prominent nucleolus and the soma. This is an artifactual shrinkage here, But another feature is present, which is seen in all ganglia, and that is the presence of these glial cells that circle around the cell body. And these are called satellite cells because they encase the entire neuron, and they're support cells essentially. So what's this stuff in the background? These are myelinated fibers, which means axons. So the term fibers is going to be used in a lot of different contexts in histology, and you've seen it before as extracellular components, elastic fibers, collagen fibers, so on. In the case of neurons, a fiber refers to part of a cell, and that means the axons. We're also going to use fibers as entire cells when we look at muscle. You already saw muscle fibers when we looked at smooth and cardiac muscle, and you will also look at them in a week or two when you look at skeletal muscle fibers. So zooming back down in magnification, you can see that there are tracts of these nerve fibers here and areas where you have many cell bodies. So if we look at these nerve fiber areas here in the DRG, we can see the centrally kind of located in the middle that's the actual axon down the center, and these are the myelin sheaths here. And in some places you may be able to locate Klefsa Schmidt-Lantermann and nodes of Ranvier. But before we do that in this specimen, I'd really like to try to do that in the T's nerve fiber, because that's the place where it's easiest to see them. So once you get it there, it becomes easier to find it in all these other specimens. So we'll do that first before we look for other examples in peripheral nerves and in these ganglia. So you looked at the multipolar and you looked at the pseudo-unipolar. The third type is a bipolar cell, and those are principally sensory receptors that you see in places like the eye and the ear, and they'll be explored a little bit in the next block rather than here in this block. However, the second type of ganglia, we already looked at the dorsal root ganglia, the second type of ganglia is the sympathetic ganglia, so let's move on to that and take a look at that. So these cells are much more small, so the soma are a lot smaller and more uniform in size than the DRGs were. The DRG cells were either fairly large or fairly small. These are all kind of medium-sized cells, and just like all neurons, they have that prominent nucleus with nucleolus right in the middle. However, as opposed to the DRG that have pretty centrally located nuclei, these sympathetic ones have a more acentric nucleus. But still, just like the other ganglia that we looked at, they're going to be surrounded by satellite cells. You can see the satellite cells circling all of these sympathetic neurons in this ganglia. So just as the first neurons that we looked at from the spinal cord, these are multipolar neurons speaking morphologically, although you don't really see their projections coming off of them very readily. You do occasionally see here and there a few kind of projections appearing to come off of them, but it's not as obvious as it was in the spinal cord neurons. Another structural difference between the sympathetic and DRG ganglia are the fibers. Here in the sympathetic, these are non-myelinated, so you don't see that kind of white space around the fibers as they're transversing through this area. The third type of ganglia is one that you saw in the prior block, and that is a parasympathetic ganglia. Those always are found within the target organ. So you looked at them in the gut tube, and that's going to be our prime example here. So as we zoom up in magnification, looking between these two layers of smooth muscle, You can see these areas where you have nervous tissue, and that's the myenteric plexus, as we learned last block. Now at high mag, we can see these neurons here, and they have their own little satellite cells surrounding them, and you can see the nucleus and so on. It doesn't have as prominent a nucleolus as the other two ganglia that we looked at. However, these are distinguished not only by their location, but by the fact that these are the smallest type of ganglia that we're going to look at. So the DRG are large and medium-sized. There's some variability. The sympathetic are more or less all medium-sized, and the parasympathetic are fairly small cells for neurons. And of course, you can also find parasympathetic ganglia in the submucosal plexus, not just the myenteric plexus. So let's move along and start examining peripheral nerve fibers and peripheral nerves as organs. We're going to start with the teased fibers. These are not sections. This is a whole mount of teased nerve fibers, in other words, the axons. Let's jump up in magnification and take a look. Here at higher magnification, you can see the blackish appearing fibers or axons. Notice that some of them are quite slender and thin, where others are a bit thicker. And you should be correlating that in your head with the speed. Remember, the diameter of the fiber is related to the speed of its transmission. The wider the diameter, the faster the transmission of the signal. So this fiber is slower than this one. Moving around in locations, you can see that you have areas where the fibers seem to end and then continue. This is a node of Ranvier, which can be defined as an interruption in the myelin sheath. So why are these things black? They were stained with something called osmium tetroxide, which turns lipids black, and myelin is mostly lipid. In some cases, you can actually see the nerve fiber in the middle and the myelin sheath outside, but here they all just appear kind of blackish. Here are some more examples of nodes of Ranvier. This is a very large diameter fiber, and this is a fairly small one, and here's a medium kind of diameter. So examples of lots of nodes of Ranvier. So what about these things called clefts of Schmidt-Lantermann? These are actual areas where the myelin sheath has been expanded and you have cytoplasm from the Schwann cell. This forms a communication between the outermost layers of the myelin sheath and the innermost, and essentially is a cytoplasmic spiral from the outside to the inside. Thus, when you look at this in longitudinal section, they appear almost like little arrowheads. So here's some examples. You can see these kind of little arrowheads here, and you can also see several of them here where you have these kind of little inward appearing areas. So these are all examples of clefts of Schmidt-Lantermann here and here and here and here. And now that we know what those look like in a fairly easy to examine specimen, we can kind of parse them out in some of the other specimens. So let's go back to the dorsal root ganglion. Here we are back onto the area where you have fiber tracts rather than the soma of the ganglion cells. Right in the dead center here, you have an example of a node of Ranvier where the two myelin sheaths meet. Running through the middle, you can see a bit of the profile of the actual axon. That's a fairly thin specimen. Other examples of axons are here and here and here, running through the middle. What about clefts of Schmidt-Lantermann? Here's a real nice example. It's these dilations here, and you can see it repeating over and over and over as this axon runs along here. Now let's look at some examples of nerves as organs. I think it's easiest to begin with the longitudinal specimen here. So let's just zoom this up a bit. Recall that peripheral nerves as organs have all the basic tissues within them. So let's start at the outer area here. The outermost covering of the nerve is a connective tissue capsule called the epineurium. And there it is. This is fibroblasts and collagen, like you would expect it to be. The nerve itself is divided into groups of fibers called fascicles. So here's a fascicle, and each one of these fascicles is surrounded by more connective tissue. The connective tissue that is between fascicles, such as this connective tissue between this fascicle down here and this one up here, is called the perineurium. Zooming in still further, you can see the wavy appearance of the nerve fibers in histological sections. All of these nuclei are glia. Well, maybe a few of them are fibroblasts as well, but the majority of them are glia, and in a peripheral nerve that would mean Schwann cells. So as you look at these, you should be aware that there's a lot of white space, and that would represent the myelin sheaths. And in this sort of H&E section, it becomes very easy to distinguish the clefts of Schmidt-Lantermann. And here's where they have that characteristic arrowhead appearance. And you're going to see them over and over and over again, all over the specimen, really. Whereas the clefts of Schmidt-Lantermann are easier to see here than in the teased fibers, conversely, the nodes of Ranvier are a little more difficult to parse out here. However, you can find them. You have to be diligent. So here's a nice example of a node of Ranvier. You can see the ending of this myelin sheath and the ending of this one as the nerve fiber itself, or the axon, traverses right through this area. There's another example over here. It's not quite as clear. So as you kind of examine this on your own, you will come across more examples, I'm sure. So let's zoom back out and take a look at the cross-section. In this particular specimen, you can see that the fascicles are widely separated by a lot of perineurium. This isn't always the case. Sometimes they're very closely applied to one another. In order to examine the cross-sectional appearance, I prefer this specimen because there's less artifact in it. So let's switch over and take a look here. Here you have two large fascicles and a very small one kind of tucked up there. So as we zoom in, this stuff on the outside, by definition, would be epineurium. This stuff between fascicles here would be perineurium. And then as you zoom up in magnification to an area where you have nice cross-section fibers, you can see a lot of detail here. These gray areas in the very center are the actual cytoplasm of the axons. They have a little bit of dark circle around them, which is the innermost portion of the Schwann cell cytoplasm, with a white ring, which is the myelin, and then pink on the outside of that, which is the majority of the Schwann cell cytoplasm. Sometimes you can see a little bit of material between these, like these guys. They might be the nuclei of Schwann cells. And of course, whether you can see it or not, there is very delicate connective tissue between all of these nerve fibers and Schwann cells, and that is called the endoneurium. So you have three CT coverings: epineurium on the outside, perineurium between fascicles, and endoneurium between the actual fibers themselves. Let's switch specimens and take a look at the toluidine blue stained nerves. These are nice because When you look at them, these are in cross-section as the other one that we just examined was. These are nice because you can see the myelin sheath very clearly outlined here. It's been stained very darkly with the axon in the center. And all of this stuff kind of between them then would be endoneurium. You can also see examples of vasculature here. Here's a nice one. You can see the RBCs inside and the smooth muscle wall of this vasculature here. What's another feature that shows up in this one that you didn't see before? You see many of these profiles appearing to have two rings. This light area between the two dark rings here is Schwann cell cytoplasm. That means you've cut right through a cleft of Schmidt-Lantermann. So you can identify clefts of Schmidt-Lantermann in these cross sections. Another thing that you can find, but I wouldn't ask on an exam, is these kind of smeared out areas where it appears that the myelin sheath is not there. And that's an example of a cross section through a node of Ranvier. I'd also like to point out this area here where you can see the really large variation in diameter. Here's some very small diameter myelinated fibers compared to very large diameter myelinated fibers. And if you look closely in these areas here, you can see very tiny fibers that are not myelinated at all, kind of tucked down here. And some of them in this area right here too. I'd also like to point out there's one here and one here, examples of, actually three, one here as well, examples of clefts of Schmidt-Lantermann being transected. Let's switch gears again and take a look at the spinal cord. This specimen for orientation has the anterior or ventral side over here on the right and the dorsal or posterior side on the left. This then would be the ventral fissure and the central canal here, which is lined by the ependymal cells. You can kind of see them here lining this cavity, and this would be filled with cerebrospinal fluid in life. Let's zero back down again. So this kind of butterfly-shaped structure here, this is the gray matter, and this is the white matter. That means these are the ventral horns, or the ventral side, I should say, and these are the dorsal side, dorsal horns. So if we want to look for the large motor neurons in the ventral horn, we want to go up to here, to this side here. And you can see, even at this medium magnification, these really, really large neurons. Remember, these are the multipolar neurons, and we looked at these ages ago, back at the very beginning of your histological experience. So let's kind of review these guys. What can we see here at high mag? You can see a very large cell. Remember, these are RBCs lined up in a row, so their diameter is about 7 to 8 micrometers. So this is a very large cell. This is the nucleus with a prominent nucleolus. And in the cytoplasm of this soma, you can see many dark purplish spots, and those are the Nissl bodies or clusters of rough endoplasmic reticulum, indicating that this is a highly synthetic cell. So the stuff, or all the materials surrounding these soma here, is called neuropile. And that's what we call the glia, and all of the processes from the glia and the neurons and the vasculature, everything outside the actual neuronal cell bodies when you're in the CNS. There are also many synapses within the neuropile. However, you really can't see them at the light microscopic level. We will see them at the electron microscopic level towards the end of the lab, we'll examine a few EMs. So moving over to the white matter area, if you zoom in on that, you can see that a lot of the axons, which are here in the center of these white areas where the myelin sheath used to reside, it's kind of processed out and there's some artifactual space now, is present. So looking at the edge of the specimen, you can see the coverings of the spinal cord. So here are the single cell, one cell layer thick of the pia mater, and then this more lacy looking arachnoid mater. These are the connective tissue coverings. The dura mater is not present in this specimen. So the second slide from the CNS is the spinal column from a rat. In this slide you can see at low mag the actual cord cut in cross section. You can see the vertebra here, which are forming, and you can see some muscle surrounding it. Recall that we looked at this when we examined cartilage and bone. And we'll probably look at it again when we look at skeletal muscle, tendon, and ligament. In any case, zeroing in on the spinal cord, you can see the very large motor neurons here. Here's the anterior fissure, the very large motor neurons of the anterior horn, and you can see the central canal here. So let's zero in and take a quick look at these motor neurons. They should be comparable to the other specimen, and indeed they are. Very large, multipolar. You can see several different projections coming off some of them. Here you can see 3, a prominent nucleus with a nucleolus in the center, very centrally located, and the really dark purpley stained Nissl bodies within the cytoplasm. So out here you can see the white matter. We can zoom a little on that. And although it's not as clear as in the other specimen, the same features are applied here, where you have the axons with the white space around them, and neuroglia here, which would be various types of glia that are in the CNS, and you'll study that in the next block. We're not going to get into that here. So these outer areas here that are contained within the meninges out here are portions of the dorsal and ventral roots. So this is the dorsal side, this is a portion of the dorsal root here. So now we're going to switch gears and take a look at the electron micrographs of the nervous tissue that we have. So this first one, N1 it's called, is an EM through a peripheral nerve. What you can see here is a very large myelinated axon here right in the middle with a many layered myelin sheath around it. This then would be the nucleus of a Schwann cell, which shows up here when you hover over that little star. The interesting thing about this is all of these little dots that are outside in the extracellular matrix. So let's zero in a little bit and look at some of the details in this specimen. So as I zoom up, you can see, hopefully, the little dots here, which are actually collagen fibrils cut in cross-section. And collectively, if I were to put a star here, and maybe over here as well, and over down in this area down here, you would say that this layer was named endoneurium. So now you've seen endoneurium for real. So I've increased the magnification to about 300% here in order to show you the many layers here in the myelin sheath. right in this area, they come out very nicely. And you can also see the details within the axon. So you can see small vesicles here. You can see larger structures, which are probably mitochondria. They look pretty blown out here. You can also see the neurofilaments. These are intermediate filaments. And then there's the small tube-like specimen structures here, which are probably microtubules. So all of that is really nicely resolved in this electron micrograph. The final thing I'd like to point out is the difference between this very large axon here and the thickness of its myelin sheath relative to medium and then smaller axons here in the bottom of this micrograph, which have a very thin myelin sheath. So again, the diameter as well as the thickness of the myelin sheath is going to affect the speed of the signal transmission. So let's switch to nerve micrograph 2, which shows a neuromuscular junction. So this can be a little bit difficult to kind of orient yourself through. Right in the center here, this is the actual axon terminal. This, on this side with all the mitochondria, is the muscle cell. What's over here? That's an endothelial cell in a capillary. And you can see a bit of a Schwann cell nucleus covering a different nerve fiber down at the bottom. So going up in magnification to about 200%, you can see here these clusters of dots which represent cross-sectioned myofilaments. So this is part of the muscle cell. And notice all of the numerous mitochondria here for energy, because muscle cells need a lot. But what you really want to look at here is this area here, where you have all of the enfolded membrane of the muscle fiber. And this is to increase surface area for the receptors that receive the neurotransmitter that is released. So this would be the synaptic cleft, although it's a little bit too dark to distinguish. You can't really resolve it at this magnification. Notice that the Schwann cell does not cover the axon terminal where it synapses with the muscle fiber. And here, this arrow is just showing one of these dense membrane folds on the muscle fiber membrane. So moving along to the third micrograph, this is actually from the CNS. I do not recall exactly where it came from, whether it's in the brain or spinal cord, probably in the brain. But in any case, you have a lot of things going on here, and probably your best example of neuropile at high magnification that you've seen yet. So over here and over here, you have two nuclei of neurons, and here's the cytoplasm of a neuron and one of the projections from it, which means it's probably a dendrite because it's very thick and you've got a lot of organelles going down into it, which happens in dendrites. And here's the cell body of this neuron, all the cytoplasm filled with many, many different organelles. This then, between it, would be projections from both neurons and glia. So this is neuropile. It's all of the projections that you see here, all of the endoneurium kind of between them, as well as vasculature and so on. So down here at the base, you can see a nice large diameter axon, which is myelinated very heavily, and then next to it, another axon which is a little less heavily myelinated. Let's look at all of these features at a little bit higher magnification. So once you're up at about 200%, you start to notice features you couldn't see down at lower magnification, such as the presence of a myelinated axon or two in this neuropile that we noticed earlier. You can also see these dark thickened areas where some of these processes contact the cell body, and that's a synapse. At fairly low mag, you can't really see the details of that synapse there. But you're going to notice several of these as you look around. So here's a dendritic synapse. This is a dendritic somatic synapse. So you will see lots of these as you look around. So we can kind of move ourselves down and look at these features. Here's a little bit of a Golgi apparatus in the cytoplasm of this neuron. There's the edge of the nucleus that we saw earlier, and here's its cytoplasm. And here's another area where we have a lot of processes of the neuropile. Here you can see more of these synapses. Right there's one. That's probably another one right there. Looks like we have a synapse between a glial cell process and maybe a dendrite. And as we move ourselves down and down and down, further and further, we see much more examples of these connections between all of these components. So here's another example right here of a dendritic somatic synapse. So that's about all there is in this micrograph. So if we go back to low mag, you can get an overview of the entire specimen and see that we've pretty much covered everything. So here's more RER at low mag, and I think that about does it for this lab. So I hope that this orientation session was useful for everyone, and please let me know if you have any questions. You can email myself or Dr. Virdes or Pratap if you prefer. We're all happy to help you.